Welcome to the Pharmacovigilance Masterclass by PharmaHealth Insights. This is Class 1, the Foundations of Drug Safety. In this class, we're going to build your pharmacovigilance foundation from the ground up. We'll understand what pharmacovigilance really means, why medicines need continuous safety monitoring, the difference between AE, ADR, and serious adverse events, and finally, we'll follow the complete journey of an individual case safety report. Whether you're a pharmacy student, a healthcare professional, or preparing for a pharmacovigilance career, this is where your journey begins. Let's begin. Pharmacovigilance. You've probably heard this word many times, maybe during your B.Pharm classes, maybe while preparing for interviews, maybe while searching for career opportunities, or perhaps you've already started working in a pharmacovigilance department. But let me ask you a simple question. What exactly is pharmacovigilance? Is it just collecting reports of side effects? Is it only a department inside pharmaceutical companies? Is it only meant for doctors? Or is it one of the most important healthcare systems ever created to protect billions of patients around the world? If you've ever been confused by terms like adverse event, adverse drug reaction, ICSR, MEDRA, signal detection, causality assessment, risk management plan, PSUR, PUBRR, or PVPI, then you've come to the right place. Because in this masterclass, we're not going to memorize definitions. We're going to understand the complete journey of pharmacovigilance. We'll explore how medicines continue to be monitored even after approval, how one patient's experience can influence healthcare decisions across the world, how pharmaceutical companies detect hidden safety signals, how regulatory agencies make critical decisions, and how pharmacovigilance professionals work silently behind the scenes to protect millions of lives every single day. Whether you're a B.Pharm student, an M.Pharm student, a Pharm.D graduate, a clinical research associate, a regulatory affairs professional, a QA or QC professional, a pharmacist, or someone preparing for a career in pharmacovigilance, this masterclass has been designed for you. By the end of this session, you won't just know the definitions. You'll understand the logic, the science, the workflow, and most importantly, you'll start thinking like a pharmacovigilance professional. So let's begin our journey. Welcome to the fascinating world of pharmacovigilance. Chapter 1. What exactly is pharmacovigilance? Imagine this: a pharmaceutical company spends nearly 15 years developing a new medicine. Thousands of scientists work on it, billions of dollars are invested. The medicine undergoes laboratory testing, animal studies, phase 1 clinical trials, phase 2, phase 3 regulatory review, manufacturing inspections. Finally, the medicine is approved. Doctors start prescribing it, patients begin taking it. Now, let me ask you something. Is the work over? Most people would immediately say, yes, after all, the medicine has already been approved. But the truth is exactly the opposite. The most important chapter of a medicine's life has just begun. Why? Because clinical trials, although scientifically rigorous, have limitations. Let's understand this with a simple example. Imagine you are testing a brand new car. Before launching it into the market, the manufacturer performs hundreds of tests: crash tests, engine performance tests, brake testing, fuel efficiency, safety checks. Everything looks perfect. Now imagine selling that car to 10 million people. Some people will drive it on smooth highways, some on rough mountain roads, some in freezing temperatures, others in deserts. Some will service it regularly, others won't. Very soon, new information starts appearing. Maybe a small component wears out faster than expected. Maybe a particular model develops a problem only after 3 years. Did the testing fail? No, the testing was excellent. But the real world is always larger than any test. Medicines are exactly the same. Clinical trials versus real life. During clinical trials, only a limited number of carefully selected volunteers participate. Every participant is closely monitored. Certain patients may not be included at all. For example, pregnant women, patients with multiple serious illnesses, people taking many different medicines simultaneously, very elderly patients, or individuals with rare medical conditions. Once the medicine is approved, everything changes. Millions of people start using it. Different age groups, different ethnicities, different lifestyles, different food habits, different climates, different genetic backgrounds, different diseases, different combinations of medicines. The medicine is now facing its biggest test, the real world. Here's an interesting question. Suppose a particular adverse reaction occurs only once in every 1 lakh patients. Will a clinical trial involving 3,000 volunteers detect it? Probably not. Not because the trial was poorly designed, simply because the event is extremely rare. But once the medicine is used by 1 crore patients, that rare reaction may suddenly appear hundreds of times. Now imagine if no one noticed. What could happen? more patients could experience the same problem. Doctors would remain unaware. Regulators would receive no information. Warnings would never be issued. Lives could be put at risk. This is exactly why pharmacovigilance exists. So, what is pharmacovigilance? Before looking at the official definition, let's understand it in plain English. Imagine you've bought a brand new smartphone. Even after it is launched, the manufacturer continues releasing software updates. Why? Because once millions of users start using the phone, new issues are discovered. Battery problems, camera bugs, security vulnerabilities, performance improvements. The company continuously monitors user feedback and improves the product. Medicines are much more important than smartphones because medicines directly affect human life. Instead of software updates, they may require updated warnings, new precautions, changes in prescribing information, restricted use, additional monitoring, or in very rare situations, withdrawal from the market. That continuous process of watching over medicines after approval is pharmacovigilance. Now, let's see the WHO definition. The World Health Organization defines pharmacovigilance as the science and activities relating to the detection, assessment, understanding, and prevention of adverse effects or any other medicine related problems. At first glance, this definition seems simple, but every single word carries tremendous meaning. Let's break it down. Science. Why does the WHO use the word science? Because pharmacovigilance is not based on guesses, it is not based on social media posts, it is not based on rumors, it is based on evidence. Every reported case is evaluated scientifically. Medical history, laboratory reports, concomitant medicines, patient characteristics, timing of the event, known pharmacology, previous reports, scientific literature. Everything is considered before drawing conclusions. A pharmacovigilance professional doesn't ask, "Do I think the medicine caused this?" Instead, they ask, "What does the evidence suggest?" That is the difference between opinion and science. Detection. The next word is detection. Imagine you're a detective. A single clue rarely solves a mystery, but multiple clues, collected carefully, begin to reveal a pattern. Pharmacovigilance works in exactly the same way. One patient develops a rash, another develops the same reaction in another city, another report arrives from another country. Individually, these reports may not seem important. Together, they may reveal a previously unknown safety issue. Detection is the art and science of identifying those patterns before they become major public health problems. Assessment. Finding a report is only the beginning. The next question is the medicine actually responsible or is something else involved? Let's consider a real life situation. A patient develops liver injury. The patient is taking 5 different medicines. They also consume alcohol regularly. They have a viral infection. Now tell me which factor caused the liver injury. It's not always obvious. This is where careful medical assessment becomes essential. Pharmacovigilance professionals review all available evidence before reaching a conclusion. Sometimes the medicine is responsible, sometimes it isn't, sometimes the answer remains uncertain until more information becomes available. And that's perfectly acceptable. Science often progresses through uncertainty before reaching clarity. Understanding, detecting, and assessing are not enough. We must also understand why something happened. Was it dose related? Was it caused by a drug interaction? Was it due to genetic factors? Did the patient have an underlying disease? Did improper storage affect the medicine? Every answer adds another piece to the puzzle. This deeper understanding helps healthcare professionals make better decisions in the future. Prevention. Finally, the most important goal of pharmacovigilance. Prevention. Imagine discovering that a medicine should not be given together with another specific drug because the combination significantly increases the risk of a serious adverse reaction. If that information reaches doctors quickly, future patients can avoid that risk. That's prevention. Sometimes prevention means updating the package insert, sometimes issuing a Dear Healthcare Professional communication, sometimes adding laboratory monitoring recommendations, sometimes restricting use in a specific population, and in rare but serious circumstances, it may even mean withdrawing the medicine from the market. The objective is not to create fear about medicines. The objective is to make medicines safer through continuous learning, the bigger picture. Here's something I'd like you to remember throughout this masterclass. No medicine is completely risk-free. Even something as common as paracetamol can be harmful if used incorrectly or in excessive doses. On the other hand, medicines used for serious diseases such as cancer may have significant side effects, yet they save countless lives because their benefits outweigh their risks. This introduces one of the most important principles in pharmacovigilance. It's not about asking whether a medicine has risks. It's about asking, do the benefits outweigh those risks for the right patient when used appropriately? That single question lies at the heart of modern drug safety. In the next chapter, we'll answer another fundamental question. If medicines are already tested before approval, why is pharmacovigilance still necessary? We'll explore the history of drug safety, the evolution of post marketing surveillance, and how lessons learned from the past shaped the pharmacovigilance systems we rely on today. So, why do we need pharmacovigilance? If medicines are already tested before approval, why continue monitoring them? In the previous chapter, we learned what pharmacovigilance is. But now let's ask an even more important question. If a medicine has already undergone years of research, laboratory studies, animal studies, and phase 1, phase 2, and phase 3 clinical trials, and has finally received approval from regulatory authorities, then why do we still need pharmacovigilance? Isn't the medicine already proven to be safe? The answer is yes and no. Let me explain. Before any medicine reaches the market, it undergoes one of the most rigorous scientific evaluation processes in healthcare. On average, developing a new medicine takes 10 to 15 years, and only a tiny fraction of molecules discovered in the laboratory ever become approved medicines. Scientists begin by identifying a potential drug candidate. Thousands of compounds are screened. Most fail long before they ever reach human testing. The few that show promise undergo laboratory experiments and preclinical studies to understand their pharmacology, toxicity, and potential risks. Only after generating sufficient evidence can a company seek permission to begin clinical trials in humans. These trials are conducted in carefully planned phases. Each phase answers a different scientific question. Phase 1, clinical trials. The first question is simple. Is the medicine reasonably safe for use in humans? Phase I studies usually involve a small number of healthy volunteers, although some medicines, such as oncology drugs, are studied in patients. Researchers evaluate how the medicine is absorbed, distributed, metabolized, and eliminated from the body. They also monitor for early side effects and determine safe dose ranges. Think of this as introducing yourself to the medicine for the very first time. Phase 2 clinical trials. Once basic safety is established, the next question becomes, does the medicine actually work? Phase 2 studies involve patients who have the disease that the medicine is intended to treat. Researchers continue monitoring safety while also evaluating effectiveness. The medicine may appear promising, but the journey is far from over. Phase 3 clinical trials. Now comes the largest and most expensive phase. Thousands of patients from multiple hospitals, sometimes across several countries, participate. The objective is to confirm that the medicine is effective and that its benefits outweigh its known risks. If the evidence is convincing, the pharmaceutical company submits all the data to regulatory authorities such as the US FDA, the European Medicines Agency, the Medicines and Healthcare Products Regulatory Agency in the United Kingdom, or the Central Drugs Standard Control Organization in India. Experts review thousands of pages of scientific data before deciding whether the medicine should be approved. This is an incredibly rigorous process. So, where is the problem? The invisible limitation. Imagine you are trying to understand the behavior of every student in India. Would interviewing 5,000 students be enough? Of course not. India has millions of students. Even if those 5,000 students were carefully selected, they could never represent every possible situation. Clinical trials work in a similar way. Although they are scientifically robust, they still involve a relatively small number of participants compared with the millions of people who will eventually use the medicine. Now consider this. Suppose a rare adverse reaction occurs in 1 out of every 100,000 patients. A phase 3 trial involving 3,000 participants is unlikely to detect it. Not because the trial was poorly designed, but simply because the event is too rare. Only after millions of patients begin using the medicine does that rare reaction become visible. This is one of the most important reasons why pharmacovigilance exists. Clinical trials cannot answer every question. Let's look at another example. Imagine a new medicine is approved today, During clinical trials, participants may have taken the medicine for 6 months or 1 year. But what happens when patients use it continuously for 5 years or 10 years? Long term safety cannot always be fully understood before approval. Now, think about another situation. During clinical trials, patients taking certain medicines together may have been excluded, but in real life, an elderly patient may take medicines for diabetes, high blood pressure, heart disease, arthritis, and cholesterol all at the same time. Drug interactions that were uncommon during clinical trials may become important in routine medical practice. The real world is different. Clinical trials are carefully controlled. Real life is not. A medicine approved after studying adults may later be prescribed to elderly patients. Different body weight, different metabolism, different organ function. A medicine may be used in populations with nutritional deficiencies, different lifestyles, different genetic backgrounds, different environmental conditions. These factors influence how medicines behave in the real world. That is why post marketing surveillance becomes essential. Let's understand this through an analogy. Imagine an aircraft manufacturer. Before launching a new aircraft, engineers perform thousands of tests: wind tunnel testing, engine testing, landing gear testing, emergency simulations. Everything appears perfect. Now, imagine thousands of flights taking place every day across different climates, different airports, different weather conditions, different maintenance teams, different passenger loads. Would the manufacturer stop monitoring the aircraft after the first commercial flight? Absolutely not. Every incident, every technical observation, every maintenance report is analyzed, not because the aircraft is unsafe, but because continuous monitoring improves safety even further. Medicines deserve exactly the same level of continuous attention. Pharmacovigilance is not looking for faults. This is one of the biggest misconceptions. Many people think pharmacovigilance exists to prove that medicines are dangerous. Nothing could be further from the truth. The objective of pharmacovigilance is not to find reasons to reject medicines. Its objective is to ensure that medicines continue to be used as safely and effectively as possible. Sometimes pharmacovigilance confirms that a medicine remains safe when used appropriately. Sometimes it identifies new precautions. Sometimes it helps doctors recognize patients who require closer monitoring. Sometimes it identifies entirely new therapeutic benefits. In every case, the goal remains the same: protect patients while preserving access to effective treatment, a real life lesson from history. Let's briefly travel back to the late 1950s. A medicine called thalidomide was widely used in several countries to help relieve nausea during pregnancy. Initially, it appeared to be effective. However, after the medicine reached routine clinical practice, physicians began reporting a rare but devastating pattern. Babies were being born with severe limb deformities. At first, these reports appeared unrelated: different hospitals, different doctors, different countries. But as more reports accumulated, scientists recognized the connection. The medicine was withdrawn in many countries, and this tragedy fundamentally changed drug regulation around the world. It highlighted one critical lesson: approval is not the end of drug safety monitoring, it is the beginning of continuous learning. This event played a major role in strengthening modern pharmacovigilance systems and post marketing surveillance. Pharmacovigilance never sleeps. Every single day, somewhere in the world, a healthcare professional reports a suspected adverse drug reaction. A patient submits an online safety report. A pharmaceutical company reviews new literature. A regulatory authority analyzes emerging safety information. A signal is investigated. A package insert is updated. A doctor changes prescribing practices. A future patient receives safer treatment. Most people never notice this process, but it happens every day, quietly, systematically, scientifically. This continuous cycle of observation, evaluation, communication, and improvement is what keeps modern medicines as safe as possible throughout their life cycle. Now, if you are preparing for an interview, this can be question for you. Why is pharmacovigilance necessary even after a medicine has been approved? A strong interview answer would be: clinical trials are conducted under controlled conditions and involve a limited number of carefully selected participants. Rare adverse reactions, long term safety concerns, drug interactions, and effects in diverse patient populations may only become evident after widespread clinical use. Pharmacovigilance provides continuous post marketing surveillance to detect, assess, understand, and prevent medicine related problems, thereby ensuring that the benefit risk profile of medicines remains favorable throughout their life cycle. By now, you should remember five important points. Approval of a medicine does not mark the end of safety evaluation. Clinical trials have inherent limitations despite being scientifically rigorous. Rare and long term adverse reactions may only become apparent after widespread use. Real world patients are far more diverse than clinical trial participants. Pharmacovigilance is a continuous process that protects patients while supporting the safe and effective use of medicines. Now that we understand why pharmacovigilance is essential, it's time to explore the language of pharmacovigilance. You'll often hear terms like adverse event, adverse drug reaction, side effect, serious adverse event, unexpected adverse reaction, and medication error. Many students and even experienced professionals use these terms interchangeably. But in pharmacovigilance, every word has a precise meaning. Let's understand them one by one with practical examples from real clinical practice. Every profession has its own vocabulary. Pilots talk about altitude, airspeed, and turbulence. Engineers discuss stress, load, and tolerance. Similarly, pharmacovigilance has its own terminology. Words like adverse event, adverse drug reaction, side effect, serious adverse event, unexpected adverse reaction, medication error, near miss, overdose, misuse, abuse, off label use, and lack of efficacy. At first, many of these terms sound similar, but in pharmacovigilance, they are not interchangeable. Each has a precise regulatory meaning. Understanding these terms correctly is essential because every safety report, every regulatory submission, and every safety decision depends on using the correct terminology. Let's begin with the most fundamental concept. What is an adverse event? Imagine that Mr. Sharma, a 52-year-old patient, visits his doctor because of high blood pressure. The doctor prescribes a new antihypertensive medicine. Mr. Sharma starts taking the medicine every morning. 3 days later, he develops a severe headache. Now, let me ask you a question. Did the medicine cause the headache? At this moment, we don't know. The headache could be related to the medicine. It could be caused by stress, it could be due to dehydration, it could be because of an underlying infection, or it could be completely unrelated. The important point is this: the headache occurred after the medicine was taken. That makes it an adverse event. Let's look at the definition. An adverse event AE is any untoward medical occurrence in a patient or clinical trial participant who has received a pharmaceutical product which does not necessarily have a causal relationship with the treatment. The phrase does not necessarily have a causal relationship is the key. An adverse event is simply something undesirable that happens after a patient receives a medicine. It does not automatically mean the medicine caused it. Think of an adverse event as an observation, not a conclusion. Let's understand this with a simple example. A patient starts taking an antibiotic on Monday. A few days later, the patient experiences an unrelated accident and visits the hospital. Did the antibiotic cause the accident? Almost certainly not. However, because the event occurred after the patient started the medicine, it would still be documented as an adverse event during a clinical trial. Why? Because researchers first document every medical occurrence and then carefully evaluate whether there is any relationship with the medicine. Good science begins with careful observation, followed by systematic evaluation. Adverse event does not mean adverse drug reaction. This is one of the most important concepts in pharmacovigilance. Every adverse drug reaction ADR is an adverse event AE. However, not every adverse event becomes an adverse drug reaction. Let's understand the difference. What is an adverse drug reaction? Let's return to Mr. Sharma. This time, instead of developing a headache, he experiences signs of an unexpected allergic response shortly after taking the medicine. The symptoms improve after the medicine is discontinued. Later, under appropriate medical supervision, the same response is observed again after the medicine is reintroduced. The available evidence now suggests a reasonable possibility that the medicine contributed to the reaction. This is no longer considered only an adverse event. It is now classified as an adverse drug reaction or ADR. According to the World Health Organization, an adverse drug reaction is a response to a medicinal product which is noxious and unintended, and for which there is at least a reasonable possibility of a causal relationship with the medicinal product. Notice the difference. An adverse event tells us that an unexpected medical occurrence happened after the medicine was taken. An adverse drug reaction indicates that there is a reasonable possibility that the medicine contributed to that occurrence. That distinction forms the foundation of pharmacovigilance. Let's take a simple analogy. Imagine you hear a fire alarm. The alarm tells you that something requires attention. It does not immediately tell you the exact reason. This is similar to an adverse event. Next, trained investigators examine the situation and determine the actual cause. Only after that investigation can conclusions be drawn. This is similar to an adverse drug reaction. Observation first, evaluation next, conclusion afterwards. Let me give you an interview tip. One of the most common pharmacovigilance interview questions is, what is the difference between an adverse event and an adverse drug reaction? A strong answer would be, an adverse event is any undesirable medical occurrence following the administration of a medicinal product, regardless of whether the medicine caused it. An adverse drug reaction is an adverse event for which there is at least a reasonable possibility that the medicinal product contributed to the event. The key concept is causality. What is a side effect? Now let's discuss another commonly misunderstood term. A side effect is an effect of a medicine that occurs in addition to its intended therapeutic effect. It may be expected, it may be unwanted. Occasionally, it may even prove beneficial. For example, an antihistamine used for allergy may cause drowsiness. A pain medicine may sometimes lead to constipation. These are examples of expected side effects that have already been identified through clinical studies. One well-known example is minoxidil. It was originally developed to treat high blood pressure. During clinical studies, researchers noticed increased hair growth in many patients. Initially, this was considered an additional pharmacological effect. Later, scientists recognized its therapeutic value. Today, topical minoxidil is widely used for managing certain types of hair loss. This illustrates how careful observation can sometimes lead to entirely new therapeutic applications. What is a serious adverse event? Another important concept is the difference between serious and severe. These words are not interchangeable. Imagine a patient develops a very intense headache that resolves completely after a few hours. The event is severe because of its intensity. However, it may not be considered serious from a regulatory perspective. Now, imagine another patient develops an allergic reaction that requires immediate medical care and observation. Although the initial symptoms may appear less intense, the event is considered serious because of its clinical significance. In pharmacovigilance, seriousness is determined using internationally accepted regulatory criteria rather than the intensity of symptoms. So, what is the regulatory criteria for seriousness? An event is generally considered serious if it results in a fatal outcome, is considered life-threatening, requires admission to a hospital, extends an existing hospital stay, leads to significant or persistent disability, is associated with a congenital anomaly or birth defect, requires medical intervention to prevent one of these important outcomes, represents another medically important condition as judged by the healthcare professional. These criteria are recognized globally and are applied consistently in regulatory reporting, severe versus serious. Let's make this distinction simple. Patient A experiences a very intense migraine but recovers at home without further medical intervention. The event is severe but not necessarily serious. Patient B develops a mild looking skin reaction. However, the healthcare team decides that immediate hospital observation is necessary because of the potential clinical significance. In this situation, the event is considered serious. Remember, severity describes intensity. Seriousness describes medical or regulatory significance. What is an unexpected adverse drug reaction? Many reactions are already described in the approved product information. Occasionally, healthcare professionals observe a reaction that is not consistent with the currently available safety information. Such reactions are called unexpected adverse drug reactions. This does not necessarily mean the reaction has never been observed before. It simply means that its nature, frequency, severity, or outcome is not consistent with the approved reference safety information. These reports receive particular attention because they may contribute to new safety knowledge. Medication error. Not every medicine related problem is caused by the medicine itself. Sometimes the issue arises because the medicine is used incorrectly. For example, a medicine may be administered in a different dose than intended, or a patient may misunderstand the dosing instructions. These situations are known as medication errors. Medication errors can occur during prescribing, transcribing, dispensing, administration, monitoring, patient use. Not every medication error leads to an adverse outcome. However, every medication error provides an opportunity to improve the medication use process and strengthen patient safety. Why correct terminology matters? Why are these definitions so important? Because pharmacovigilance depends on clear and accurate communication. Correct classification helps pharmaceutical companies, healthcare professionals, and regulatory authorities evaluate safety information consistently across the world. Using the correct terminology supports accurate reporting, meaningful analysis, and informed regulatory decisions. Some interview questions you should now be able to answer confidently. One, it's there one. What is the difference between an adverse event and an adverse drug reaction? Focus on the concept of causality. 2. Is every serious event severe? No. Seriousness is based on regulatory criteria. Severity refers to intensity. 3. Can a medication error occur without an adverse event? Yes. A medication error may be identified and corrected before it affects the patient. These situations are often called near misses and are valuable learning opportunities for improving patient safety. Let's summarize. An adverse event is any undesirable medical occurrence after the use of a medicine, regardless of causality. An adverse drug reaction is an event where there is a reasonable possibility that the medicine contributed to the occurrence. A side effect is an additional pharmacological effect that is often expected and already recognized. Seriousness is determined by regulatory criteria, while severity describes intensity. Unexpected adverse drug reactions differ from the approved safety information. Medication errors are preventable events related to the medication use process and are important components of patient safety. We've now learned the language of pharmacovigilance, but here's an interesting question. Imagine a healthcare professional in Bengaluru documents a suspected adverse drug reaction. How does that single observation travel from a hospital to a pharmaceutical company to national regulatory authorities and eventually become part of the global pharmacovigilance system? That journey begins with one of the most important documents in pharmacovigilance, the Individual Case Safety Report or ICSR. Next, we'll follow the complete journey of a safety report and see how a single observation can contribute to improving medicine safety worldwide. Imagine you're working as a pharmacovigilance associate in a pharmaceutical company. It's 9 o'clock on a Monday morning. You log into your safety database. Several new cases are waiting for review. Each case represents a real patient, a real healthcare professional, a real medical event. Some cases may ultimately confirm known safety information. Others may reveal entirely new knowledge about a medicine. Every case matters. Which este. Who can report an adverse event? Many people believe that only doctors can report adverse events. That isn't true. Depending on the country's reporting system and applicable regulations, safety reports may originate from many different sources. These include physicians, pharmacists, nurses, dentists, clinical trial investigators, pharmaceutical companies, patients, caregivers, consumers. Today, many countries also provide online reporting systems allowing patients and healthcare professionals to report suspected adverse reactions directly. Every report contributes to the growing body of medicine safety information. Now, here's an important concept. Not every email, phone call, or complaint automatically becomes a valid ICSR. International pharmacovigilance guidelines require four minimum elements. Without these, the report cannot usually be processed as a valid individual case safety report. Let's understand them one by one. The report must relate to an identifiable patient. The patient's complete identity is not required, and patient confidentiality must always be protected. There should be sufficient information to establish that the patient is a real individual. For example, initials, age group, gender, date of birth, patient identification number. This helps avoid duplicate reporting while protecting privacy. The second requirement is an identifiable reporter. Someone must have provided the information. The reporter may be a physician, a pharmacist, a nurse, a patient, or another healthcare professional. Basic contact information allows follow-up if additional details become necessary. Every ICSR must identify at least one suspected medicine. Important information includes brand name, generic name, strength, dosage, form, dose, route of administration, and frequency. Batch number, when available, therapy start date, therapy stop date. The more complete the information, the better the quality of the report. Finally, the report must describe the medical event. Examples may include skin rash, dizziness, abnormal laboratory findings, liver enzyme elevation, allergic reaction, fever, gastrointestinal symptoms. Remember, the event is documented first. Whether the medicine actually caused it is evaluated later. Imagine receiving this email that says a medicine caused a problem. Can the pharmacovigilance department process this report? Not yet. Which patient, which medicine, what happened, who is reporting it? Without these essential details, meaningful evaluation becomes impossible. That's why every pharmacovigilance professional carefully checks whether the minimum information required for a valid ICSR is available. Now let's continue following Mrs. Priya's case. The report reaches the pharmacovigilance department. A drug safety associate receives it. The first responsibility is case intake. This means reviewing the information to determine whether a valid ICSR exists. If essential information is missing, the associate may contact the reporter to request clarification. Perhaps the exact date is missing, perhaps the medicine name is incomplete, perhaps laboratory results become available later. This process is called follow-up. One of the most important principles in pharmacovigilance is this: the quality of an ICSR depends on the quality of the information collected. Once the report is considered valid, formal case processing begins. The pharmacovigilance professional carefully reviews every detail: patient demographics, medical history, concomitant medications, dose dates, laboratory findings, reporter comments, clinical outcome. Everything is evaluated systematically. Nothing is ignored, nothing is assumed. Every case is handled according to established procedures and regulatory requirements. Imagine a patient develops a rash before starting a medicine. Could the medicine have caused it? Probably not. Now imagine the rash begins 2 days after starting the medicine. Now a possible temporal relationship exists. Timing is one of the most important clues in pharmacovigilance. That's why accurate dates are so valuable. During case processing, another important decision is made. Does this report meet the criteria for seriousness? Remember the concepts we learned in the previous chapter: hospital admission, life threatening events, significant disability, congenital anomaly, medically important conditions. Correct classification determines regulatory reporting timelines. This is why accurate assessment is so important. Next comes another question: is this reaction already described in the approved product information? If yes, it may be classified as expected. If not, additional evaluation may be required because the report could represent emerging safety information. Unexpected reports often receive particular regulatory attention. Why every ICSR matters. Some people assume that one report cannot make a difference. History has repeatedly shown otherwise. Many important safety signals began with only a handful of carefully documented reports. One report leads to another. Patterns begin to emerge. Scientists investigate. Regulators review the evidence. Recommendations evolve. Patient safety improves. Every ICSR is therefore much more than a form. It is a contribution to global medical knowledge. Interview Corner. A common interview question is: what are the four minimum criteria for a valid ICSR? A strong answer is a valid individual case. Safety report requires four minimum elements: an identifiable patient, an identifiable reporter, a suspected medicinal product, and a suspected adverse event. Another common question is: why is follow-up important? Because additional clinical information improves case quality, supports better causality assessment, and strengthens the scientific value of the report. Key takeaways. Let's summarize what we've learned. An ICSR tells the story of one patient's suspected medicine related event. Every valid ICSR requires four minimum elements: an identifiable patient, an identifiable reporter, a suspected medicinal product, a suspected adverse event. High quality information leads to high quality safety evaluation. Follow up is an essential part of pharmacovigilance. Every ICSR contributes to the global understanding of medicine safety. And with that, we've reached the end of class 1 of the Pharmacovigilance Masterclass. Let's quickly look at what we've built. We started by understanding what pharmacovigilance actually means. We then explored why safety monitoring must continue even after a medicine receives regulatory approval. We learned the critical difference between an adverse event, an adverse drug reaction, a side effect, seriousness and severity, unexpected reactions, and medication errors. And finally, we followed the journey of an individual case safety report from the initial report through the beginning of the pharmacovigilance process. These are not just definitions, they are the foundation on which the entire pharmacovigilance system is built. But we've only opened the door because in the next class, we're going deeper. How do we standardize millions of medical terms from around the world? How do we identify a safety signal hidden inside millions of reports? How do we determine whether a medicine actually caused an adverse event? And what really happens inside a pharmacovigilance department when a case arrives? That's where the science becomes even more interesting. I'll see you in class 2 of the Pharmacovigilance Masterclass. Until then, keep learning, keep questioning, and remember behind every safety report is a patient whose experience can help make medicines safer for someone else.